Welcome to this brief presentation of this tool. Thank you to the organizers for having me presenting this and for giving you the chance to know about these works that were done by me and others in different institutes. So my name is Arnau Montagud. I'm the head of the Systems Biology Modeling Lab at I2SysBio, which stands for Institute for Integrative Systems Biology. That is mixed institute between CSIC and University of Valencia. So yeah, let's get this started. Yes. So I plan to divide this presentation in three blocks. One would be a short primer on Boolean modeling, so that we all know what we're talking about when we talk about Boolean modeling. Then how we can personalize models using this profile tool using on top of these Boolean models. And then some examples of personalized models in different databases with publicly available data. So some of you may note that Boolean logic is a convenient way to model regulatory and signal networks. Usually, when questions that are well fit to work with Boolean modeling are usually questions that are qualitative, or in which data is discrete, or at least it's semi-quantitative, like we can have in expression data, in the sense that we don't have absolute values, and also there is no information about time, and there is very few details or no details about the precise biochemical reactions. So typically, this is what we have in most of molecular and cell biology, in which what we know is that, for instance, node A is activated by C and is inhibited by B, but we don't know the rates of activation, we don't know the rates of inhibition, we don't know if it's allosteric inhibition, if it's sympathetic, etc. So when in these cases, it's good to use Boolean model because it gives you a very easy way to build mathematical constructs in which you can perturb these signaling pathways or these irregulatory networks. So usually this starts with a regulatory graph. So you just draw what are the activations and inhibitions between A, B, and C. Then you translate this regulatory graph into logical rules. So you know that the activation of A depends on the non-presence of B and the presence of C, and so on and so forth. So in this case, in these formulas, each variable takes two states, inactive or active. And then you gather the activations and inhibitions using Boolean logic. So you have connectors, and then of course the logic depends on the incoming arrows of the regulatory graph. So of course we'll know that things in biology are not Boolean, and there is no such thing as inactivity or activity. So everything is dynamic and extremely elastic. But still, this is an abstraction of the activity levels of these proteins, of these genes, of these transcription factors, whatever. And then, of course, those are linked by these signed interactions. So the way when we talk about simulation of Boolean models, the way we simulate this is to find the attractors of the system, which means where does this model end? I mean, if you evolve this model, where does it end in its dynamic? So these attractors are usually two kinds of attractors, stable states or cyclic attractors. So in this case, in this very, very simple toy model, we have two solutions. One is the nil state, so 0, 0, 0. So whenever C is not active, then eventually B will inactivate A, which is what you have here. Here the value for A, B, and C. So if C is 0, what you end up is with everything goes to 0 because B inactivates A. And the other solution in this toy model is when C is active, which means that whenever B inhibits A, then C will activate A. So in this case, what we have is four states that are cycling one to another. And so usually in biology, in our modeling, we tend to work mostly with stable states, so with what we call phenotypes, mean migration, proliferation, apoptosis, things like that. And we usually tend to don't consider or to veer off cyclic attractor, even though there are some Boolean models that are basically built around these cyclic attractors. So yeah, in our case, stable states will mean self-fates, which is a way of understanding different phenotypes. Again, abstraction of phenotypes. So what insights can we get out of Boolean models? So for instance, usually the questions are, what are the solutions of the models? For instance, we have a set of inputs, we have a set of outputs, and we now want to know what combination of inputs and maybe perturbations in the different pathways will veer some trajectories out of survival and go into necrosis or to apoptosis. Or for instance, what are the most important nodes in this network, of how robust or sensitive these networks are. So here on the left, what you have is an example of a TNF response on the different cell phase that you can get from a 2010 paper by Institut Curie. And yeah, so this model, usually when I talk about studying its attractors, we do this in what it's called a truth table, which is the combinations of activation and inactivation of the different inputs and internal nodes that gives you the values of the different outputs. So in this case, white is 0, black is 1, and in columns you have the different attractors, and in the different lines what you have is the different values of the nodes. So for instance, if FAD is active, which is these 2 here, this 4 here, and this 4 here, this means that the possible solutions of the system are reduced in to those attractors where FAD needs to be 1. And you can see that most of them are apoptotic and also necrotic. There are some of them that force, there are some inputs or some perturbations inside the model that force the system to not have some of these solutions. For instance, when ATP is 1, what you see is that you don't have necrosis. You have the other 3 kinds of attractors. So this is the way that we usually simulate with Boolean model. And you can see that you have high complexity of the truth table depending on the rather simple or reduced model node numbers in the model that you have on the left. So other examples of Boolean models or logical models is, for instance, T-cell signaling in an adaptive immune response. So here what you have is activation and inhibition of how the different T-cell is differentiating. And then one of the examples of cyclic attractors that are in Boolean models and are made in order to have these cyclic attractors is this cell cycle control next checkpoint. So here what you have is that whenever the cyclins are activating and inhibiting each other, what you have is like this wave of activation that goes through the different cell states. So in this case, you don't have a stable state. What you have is cyclic. They are cycling one after the other and reproducing what we see in the experiments in molecular and biochemical biology. So several years ago, a group at Institut Curie, headed by Emmanuel Barillo, decided to have something more complex than Boolean modeling, but not as complex as ordinary differential equation. So they came up with this tool called MABOS, in which what we try to have is a stochastic Boolean modeling simulations. And in order to do this, what we did is, or what they did is to work on this state transition graph. So this is where the model is heading, let's say. And for each Boolean state, they associated a probability. So now we have a rate of change associated with each transition. So node A, B, and C will have different rates of activation and rates of deactivation. Okay, so you have a rate to go up and a rate to go down from 0 to 1 and from 1 to 0. And then what we do is that we're using MABOS is that you play with thousands of trajectories depending on different initial states. You let the trajectories run, and then the trajectories will land in one or several of these phenotypes of these stable states. So these probabilities are important whenever you have this kind of paths in which you have like a Y cross path in which you need to choose to go left or right. And then you will evaluate the different probabilities, and some of the trajectories will go to one place and the other will go to another. And in this case, you can study stochastically these models, and you can study probabilities, and you can even have some pseudo time of this. And you can also study perturbations in a probabilistic, and also you can have those such experiments as we will see in a minute. So now on the left is what we have, let's say, with the classic Boolean modeling, in which you have that whenever you have inputs or whenever you have perturbations, you have that all the states of the model go to one place, to go to one state or another. In the case on the right, which is using MABOS, what you have is that you have different probabilities that are shifting depending on the perturbations. For instance, in this case, is a change on initial conditions. On the top, you have all input random initial conditions, and then you see that the percentage of naive is 47%, the percentage of apoptosis is 34%, etc. And then whenever you change these initial conditions, you see that this 34% of apoptosis goes to almost 70% of apoptosis. So in this case, we're changing the probability of reaching the apoptosis to almost twice as high as previously done. And this you can do with the initial conditions, and also you can do with internal nodes. You can perturb the node and say this node will always have zero value no matter what its activators are, and then you can study these perturbations semi-quantitatively, at least relating it to some wild type or to some basal state. So this is the framework on top of which Profile works. So Profile, what it does is that it takes this generic logical model that you can have of breast cancer, of prostate cancer, or whatever, and then taking different omics data from different patients or cell lines or whatever. What we do is that we personalize these models so that we have different modeling framework variables, like node states, initial conditions, and the transition rate. We tally them with different kind of data types. And these can be copy number alterations, they can be mutations that are easily Boolean, so we can convert them to zeros and ones. Or you can also have expression data. So for instance, RNA or proteins that we can assume that they are easily continuous and that we can tag to some of those variables. So from, let's say, a given breast cancer model, we can have different patient-specific or at least dataset-specific models. And this we have done, for instance, in these cases with different cell lines, and also we have done with patients, as we will see in a couple of slides. So first, as we are bioinformaticians, we like a lot to wrangle data and to like put data into boxes. So one of the things that we did is to take in a gene expression across all the different data sets that we were personalizing. We try to organize these genes into 3 boxes of genes, let's say bimodal genes, unimodal genes, and zero-inflated genes. And in order to do that, we have different admissibility tests, which is basically quality control. We have a bimodal test, and we have also a zero-inflated test. So depending on the values of these 3 tests, we will consider a gene, and we will assign the gene to these different bimodal, zero-inflated, and unimodal genes. Of course, we could complicate this organization much more, but we wanted it to keep it simple, and also that it makes sense with what we are doing down the road with these genes. So basically, what we want to do is each one of those gene sets to identify a binarized distribution, so to assign 0 or 1 of a patient on that gene, and also to have a normalized distribution, so continuous values between 0 and 1. So if, for instance, with the bimodal genes, in order to have this binarized distribution, what we did is GMM2 components in which we try to decompose this bimodal into the two Gaussians, so that all the patients that have values in here will be assigned a 0 value for these genes, and all the patients that have value here will be assigned a 1 in the genes. For the unimodal genes, this was done using just outliers. So we identify the different outliers on the low part and on the high part, and we assign zeros and ones to those. And on zero inflated, we just assign ones, as if we assign zeros, basically the model just stopped working because there were so many values with 0 in this case. And similarly, what we did for the continuous values. So on the continuous values, on the bimodal genes, we basically binarized them, which was not very useful later on. But for instance, for the unimodal genes, what we did was to, let's say, twist or extend the values of the expression between 0 and 1. So what we have is that we have continuous values from zero, and so on with a linear normalization for the zero inflated genes. And we did this in order to assign the value of a gene in a patient to a given variable in the frame. In this case, we're playing with 3 different variables: initial conditions, node states, and transition rates. So looking at the Waddington landscape that was published many years ago, a way to conceptualizing thinking about this is that the initial conditions is where you drop the little marble here. So that initial conditions can be on the top of the mountain, can be on the valley, can be in this valley, etc. And depending on these initial conditions, the model will follow one trajectory or another. If we change node states or transition rates, what we're doing is that we're changing this landscape. So we're going down, beneath this landscape, and we're pulling and we're pushing the different columns that this landscape has. So we can convert a mountain into a valley, and we can convert a valley into a mountain. And in this, changing the node state has much more, let's say, transformative power than changing the transition rates. But there's always a question when we do this personalization: what data or which data to use in which variable, and how many data types to include into our personalization. One tends to think that the more data, the better, but using other kind of validation, you realize that that is not always the case. So, for instance, in breast cancer, what we did was to assign different data types to node states and to transition rates. So it's these two columns here, and we looked at what was the let's say the spread of the apoptosis and proliferation value of the model using these personalized values for all the in this case it was almost 2,000 patients. And you see that the different spreads of the scores, the different distributions change quite a lot depending on what data you use. Of course, if you use mutation data are almost discrete values, but if you use just, for instance, RNA, you see that there's a continuous values of this score. Then the problem is, okay, which one is better? What combination here is better in order to personalize my models? And for this, what we usually use is two things. One is hallmarks. So we remove from the hallmarks if the gene is considered in personalization. And for the rest of the gene, we compare what is the value of this apoptosis score with this hallmark. And we kind of have a correlation of, the better the hallmark, the better the correlation means that my model is capturing better this apoptosis hallmark in my model, and the same for proliferation. In the case of breast cancer, we also had for these patients, we also have a clinical prognostic index, which is called the Nottingham Prognostic Index, or NPI. And then again, we study the correlation of the NPI between what we were simulating in the proliferation of the model with the NPI score. And you realize that is not in the one that is using most of the data where we are the best correlation. Usually is in this case of the apoptosis is just including RNA, and in the case of proliferation is including mutations on RNA. And also with this clinical index is also the combination of mutations or RNA, meaning that the copy number alteration with this specific model doesn't include better data for the personalization process. This doesn't mean that the CNA is not useful. It means that for the abstraction that represents the model, the copy number alteration is not increasing the accuracy of the mutations and the RNA. And this we did similarly with prostate cancer, in which in this case we didn't have any index for any prognostic value for these patients. But what we had is that we distributed them at the different Gleason scores, so the subtypes of the patient, 1 meaning mostly benign and 5 meaning very, very aggressive. And what we wanted to see is that in looking at the proliferation score, the more aggressive patients would have higher proliferation scores. And then we ended up thinking that maybe mutation RNA was much better than including the copy number alterations. Again, this is looking at the problem through the lens of the model. With other models, this will change for sure. So a few examples of what we can do with this personalization, looking at the different prostate cell lines, is to study their values at the different phenotypes. So we can see that comparing the wild type with the different cell lines and personalizing them with a mixed data, we see that they have different values of the different scores in these phenotypes. And the same happens, for instance, in this case, what we focused in one cell line, in this case LNCAP, and we wanted to do all the mutants of these LNCAP, the LNCAP models. Here you have all the single and double mutants compared to the wild type, and then we have values for all the phenotypes. And then what we do is do a PCA on the matrix of all these mutants. And here what you can see is the distance of the different mutants in regards with the wild type. So looking at this graph is a bit complex, and then of course you have the PCA flattening of it, so it's a bit hard to interpret anything. But what we usually do is that we look at let's say one of those variables of the PCA values, and then we look at distance of some of the mutants regarding to this wild type. So you can identify mutants that have no invasion phenotype, which is what you would like in order for a drug to take effect. And also you have mutants that increase their invasion probability. Usually is because you knock out some inhibiting factor, and then the model can run loose in the invasion phenotype. So likely with this, what you can do is that having these dosage experiments that I was saying before out of MABOS, what you can do is to have increasingly inhibitory effects of the model. So you can work as if you were working with different drugs that would inhibit the activity of a given protein. And this you can do for, in this case, is two different inhibited nodes, AKT and beta-catenin, and you can look at what is their effect in the different phenotypes, and what is their effect, of course, of different growth conditions. So you can see, for instance, that the inhibition of beta-catenin is mostly affecting invasion, as you could have all guessed, but then this is quite dependent on the growth condition that we're using. So in fact, if it's AR is active, this inhibition does not take effect. Okay, and this you can do for one drug, but also you can do for combinations of drugs. So this is what we did on this paper on prostate cancer, in which we looked at the different inhibitions of MICMAX and ERK, and also PI3K and heat shock proteins. And we found out that those two combinations had a Bliss score that was low, in the sense that they could have synergies between the dosages of these drugs. So this we talked with a collaborator, experimental collaborators, and we convinced them to perform some experiments, and this is what they did. They were not very convinced to do the HSP experiments, but in fact, what they found out is that some of the drugs that targeted HSP had greater effect than the drugs that were targeting PH3K. So this was good. Unfortunately, we don't have the data for the combined inhibition of both to see if we have this synergy. But yeah, I'm very happy to talk with anyone that would like to do these experiments. So this was for cell lines, and then we did also this for different patients from different databases, for instance, the CGA and Metrabric in prostate and in breast cancer, and we found out similar things. That for instance, when the Gleason group was increasing, the different models of this Gleason group were moving away from DNA repair and closer to proliferation and migration, which gives a taste of that the model is capturing well this aggressivity of these different patients. And then also one of the things that we wanted to test is if we could fish out candidates to be inhibited at the different groups and subgroups, and if those candidates were different. And what we have here is, in this panel we have all the patients confounded, and then we have the Gleason group 1, 2, 3, 4, and then 5. And you can see that there is some common, let's say, druggable targets or wishful druggable targets like AKT or like 6-cyclin D or SPOP, etc. But there are some of them that are a bit different from one to the other. For instance, MICMAX only comes in the picture starting with Gleason 2-3, and then in Gleason 5 is like the tough hit to be inhibited if you don't want proliferation in these tumors. So that was for, let's say, for targeting different proteins that are captured in the model. And one of the things that we were lucky enough to do with using the Metrabric clinical data was to look at survival data. So here the idea is that you have the 2000 models for the Metrabric database. You run the 2000 models, and then you know what is their score in terms of proliferation and apoptosis. And you can look at what is the survival that those patients had. And what we saw is that the proliferation and the apoptosis score, using a cutoff, differentiated well between the let's say the ones that survive the more and the ones that survive the less. Ideally, what you want to, ideally, in those kinds of cancers, what you want is to have low proliferation and to have high apoptosis. And in fact, the model captures well this. And if you look at the combinations of low proliferation and high apoptosis, those are the patients that survive the most in the metabolic database. So yeah, this was using the outputs of the simulation and just cross-validating it with the survival data. So yeah, so this I said I would split the talk into three blocks, but now maybe I have just a few minutes just to give some spoilers of if you're lucky enough to come to the talk in March in Madrid, just so you know that this personalization of models, we have included it into this multi-scale agent-based modeling framework that we're using. We're using IC50 curves in order to connect whatever is happening outside to whatever is happening inside of these cells. And then what we can get is these patient-specific drug response curves, depending on which drug you add and to which patient you are affected, or to which cell line you're affecting. And this, we can also do these in combinations. So we also have this combination of drugs using the agents, using the multi-scale tools. And we can also optimize drug regimes in vitro, in which we can tally different experimental data using our simulations, and also using different optimization techniques. And lately, yeah, so we can do that. I wouldn't say high throughput, but multi throughput with different drugs. And we are also integrating different single cell and also spatial data, omics data, into this personalization framework. So yeah, I think that's it. So I left here the resources in case you guys are interested into tinkering with this. And yeah, thank you very much for having me, and thank you very much for your attention today.